As we dream of colonizing distant worlds, one question consistently guides our search. Is there water? Water is not just a requirement for life. It is life's medium. Every cell depends on it, not only to survive but to function properly. Within living cells, water movement isn't random. It's governed by a process called osmosis. Osmosis is the passive movement of water molecules across a semi-permeable membrane, and it allows only certain molecules or ions to pass through but not the others. It is always from a region of low solute concentration to a region of high solute concentration. This movement is vital. It helps cells maintain their shape, like how plant leaves stay firm and upright when well watered, but wilt when water is lost. Osmosis also plays a key role in how cells absorb nutrients and respond to changes in their surroundings. Let's take a quick quiz. Can you identify which of the following examples actually involve osmosis? A dishwashing sponge soaking up water, salted or marinated vegetables becoming wet and releasing juice, aromatherapy candles filling the room with scent. Remember, osmosis specifically refers to the passive movement of water across a semi-permeable membrane. So, which of these involves that kind of movement? Take a moment to think. Here are the correct answers. Salted veggies turning wet. This involves water moving through membrane driven by differences in concentration, which is the hallmark of osmosis. The sponge is an example of absorption and the scent is an example of diffusion. So now we have seen where osmosis happens in everyday life. Let's observe it under the microscope. For this experiment you will need glass slides, a marker, Tradescantia leaves, water, some sugar, and of course a microscope. Now let's get started with the experiment. So, I got 6 test tubes here. I'll number them from 1 to 6. In tube 1, I'm adding 1 full spoon of sugar. Tube 2 gets half a spoon. In tube 3, I'm adding quarter spoon. Tube 4 gets about half of that. Tube 5 gets just a pinch. And in tube 6, I'm adding no sugar at all. That one is our control. Now, I'll pour water into each of these tubes until the 25 ml mark. I'm closing the lids and I'm giving them a good shake till the sugar dissolves completely. And that's it. I have 6 sugar solutions, each a little less concentrated than the last. Now before we go ahead, take a second to think about what is happening here. I'm going to place a piece of Tradescantia leaf epidermis, so basically a thin layer of plant cells, into each of these solutions. So what do you think will happen to the cells in these different sugar solutions? If you know osmosis, the idea is simple. Water always moves from where it's less concentrated with solute, which is more watery, to where it's more concentrated with solutes, which is less watery. So, if there's more sugar outside the cell, water will move out of the cell, from the vacuole through the cell membrane into the sugar solution. And when that happens, the cell starts to shrink. But remember, the cell sap inside already has some sugars and solutes too. So the big question here is, how much sugar outside is just enough for the cell to stay normal? Let's find that out. I've peeled a Tradescantia leaf and made a fresh microscope slide. Under the microscope, the cells look beautiful, neat, hexagonal with pink vacuoles inside. Now I'll add the first solution, tube 1, to the slide and observe. the same beautiful hexagonal vacuole. Next, I'll wait for a couple of minutes. Look closely. Do you see it? The pink vacuoles are starting to shrink away from the cell wall. After about 10 minutes, it's clear. This solution is too concentrated. Most of the cells show shrunk vacuoles. Then I'll repeat the same steps with tube 2, tube 3, tube 4, tube 5 and tube 6, one by one, and record all my observations. Now when you compare them, tube 4 shows cells that look relatively healthy, less shrunken, and tube 5 and 6 show healthy cells, no shrinkage. So, somewhere between tube 4 and 5, the concentration of solutes inside and outside the cell is balanced. The cells are said to be in an isotonic solution. Here are a few questions for you. Based on your observations, do you think that the plant cell wall, remember, I'm not talking about cell membrane, the plant cell wall is also semi-permeable? Why and why not? If you replace sugar with salt, will you still observe similar results? We now know that the osmosis is a vital process. But can you think of a commercial application of osmosis? I'll leave you with these questions and move to discuss classical example of osmosis with you. So, water is a medium of life. It must enter and leave plants for biological water cycle to complete. Water enters a plant through its roots, more specifically through special cells called root hair cells. These tiny projections increase the surface area of the root, making it easier to absorb nutrition and water from the soil. Water moves into these cells by osmosis, just like we saw in our experiment. Once inside, the water fills up the vacuole, causing it to push outwards against the cell wall. This creates what we call turgor pressure. It's a kind of internal water pressure which helps the cell maintain its shape. It also helps move water to neighboring cells. Eventually, the water reaches the xylem, which acts as tiny pipelines, carrying the water upward against gravity all the way to the top of the plant. So, xylem is actually a network of long hollow tubes made up of dead cells called tracheids and vessels. Now pause and think for a moment. Can you think of any other example where liquid travels upward, but without pump, pulley or any external energy? Let's take a simple example. The Diwali diya. The cotton wick inside the diya is made up of thin fibers which absorb and carry oil. Only the base of the wick touches the oil, but the flame burns at the tip. Can you guess what in plant works like the oil, the wick? Does it also involve absorption and capillary action? Yes, the oil is like water in the soil, the wick is like the root hair cells and the xylem. The process involved is osmosis and capillary action. As the flame burns, it uses up oil at the top, so more oil rises from below. This occurs naturally. There is no battery or pump involved. It's just capillary action and the cohesion adhesion forces of the liquid. In plants too, water is pulled upward in a very similar way. So, what exactly is this upward pull and what role do leaves and stomata play? That's what we'll explore next when we'll observe stomata under the microscope and dig deeper into the process of transpiration. Stay tuned.